So, our final presentation, we're moving from one yogurt to the next. We have Sylvie Turgeon and Danielle Saint-Gelais, one from the University of Laval and the other from Agriculture and Agri-Food Canada. They're going to be talking about stirred yogurt quality, stability, and rheological properties. Ms. Turgeon. So you'll see Danielle is seated, but will be there to support me in the questions. We agreed that I would be the spokesperson for the presentation. So briefly, let me talk about context. When we started the project, after discussions with industrial partners, we quickly realized, and when we looked at the literature, that a lot of work had been done on stirred yogurt, but it didn't reproduce what we found in industry. There were a lot of studies on firm yogurt, a lot of studies in which the firm yogurts were stirred with a spoon or they used syringes, but not the kind of things that we would actually find in an industrial setting. So we developed the project on this basis to make the connection between yogurt composition, mechanical stress, shearing during the process. And the final characteristic, we know that consumers, when they eat a yogurt, like a certain firmly, smoothness and unctuousness, but they want stability too. There are various ways to control these various properties of stirred yogurt, of the composition of milk, the presence of protein. Whoops, the slides are starting to advance on their own again. We'll see if it will stay put. Okay, if we play on the ratio of casein and whey protein, we can get the desired qualities. There's the presence of fat and solid, and there will be an impact on the properties of yogurt. The presence of stabilizers, which are widely used in North America, was a significant factor as well, which we didn't study as part of our project because, depending on the formulation, there's a question of competition. Different companies use different stabilizers. The starter can play an important role. In recent years, there was a program at Novalait to look at PS productive strains, which we didn't do in our case. There are a whole series of manufacturing processes that can have an effect. In terms of there's a process whereby the casein results associated with whey proteins are fermented. Then there are mechanical stress through pumping and smoothing and stirring that produce microgels that will come together during storage. As part of our project, we were interested here we see the various stages of processing, and then we studied the effects of stirring, cooling, smoothing, and storage to and what I'm presenting are the results that were got by our various students, that the main results. But there are 3 posters for this project, 2 by Audrey Gibert, another by Valérie Guénard-Lampron, who is a doctor and doing an internship in France. Laurier-Frioux will be presenting that poster today. So a first study looked at the stages 6 and 7. The impact of temperature and cooling. We used a yogurt that was standardized at 14% total solid, 4% protein. The CNPS ratio and zero fat. This was used throughout, so our results could be comparable. So in terms of our parameters, we had stirring 5 or 10 minutes. We use smoothing. Stirring is here. And when you see a little circle, that's where sampling was done. The sample was cooled and the properties were measured the next day after 24 hours. These are not inline measurements. It's important to remember that. So what we studied was smoothing, either immediately after fermentation or before cooling, or after at 20 degrees Celsius. And we compared two cooling apparatus, a plate heat exchanger or a tubular heat exchanger. And we have a configuration here that shows that we have smoothed right after fermentation and cooling, or we pre-smooth before stirring and after pumping and pre-smoothing, after cooling and after smoothing in configuration 2. So in terms of syneresis, firmness and viscosity, we have the two, whether we smoothed at 28 or 20 degrees Celsius. In the two configurations, there's no difference in syneresis of the product a day after processing. Fermentation, however, smoothing is important to give a certain or a high firmness at 38, but if it's done at 20 degrees Celsius, there's a great difference in the firmness here at the time of cooling. But ultimately, the yogurts are slightly different, but less so. I just want to show the viscosity results. Now, here, smoothing at 38 or smoothing at 20 gave us values that were ultimately similar. So when we cooled, you can see here at 38, there's a difference in viscosity, but it recovers it through smoothing at 20 to provide similar properties at the outcome, at the end. Now, we had two extremes, 20 versus 38, and we thought it would be useful to look at smaller temperature ranges, 10 to 35, with 5-degree increments, using the same formulation. In all cases, what we were comparing was smoothing, which appeared at the same time after the pre-smoothing stage. So, what I'm presenting here is syneresis and firmness. The poster shows consistency and viscosity as well. Since we used various temperatures, it was possible to model the results. And I'm showing you here for the first time what happens on the basis of storage. For syneresis, there's a considerable impact of temperature when the product is smooth. And the longer it remains at high temperature, the higher the syneresis is. But there's not a great change based on storage time. However, in the case of firmness, measured by texture, in the samples, we can see there is a bell curve. There's a temperature at which we can have increased firmness, but up to, if we smooth at 38, firmness goes down again after being high at 30. Now, you can see more details on the stage, but there's no temperature that enables us to optimize all properties. You have to decide which are the most important for you. But contrary to syneresis, where there was no difference based on storage time, here we see that there is an important increase in firmness and lessening of the effects of smoothing temperature. So it is important to consider smoothing temperature and storage time to get a specific property. Now, here's what I was explaining earlier. If you do a relative sensitivity study on syneresis over 1 day or 1 degree in smoothing temperature, it's temperature that has the most impact. But if we looked at the flow time results, storage time has a major impact on that particular property. So this enables us to model and determine which are the most important factors. What is the impact of composition? As part of another student's product, Noémie Lucier, we looked at the impact of fat content, cooling system, and fermentation rate using our sample yogurt, our control yogurt, but also with other yogurts where we had different fat contents. And here are the main results. Again, in terms of syneresis, when you increase the fat content beyond 1.3% of fat, we have a significant drop of syneresis in the product. Similarly, for firmness, when you go beyond a certain concentration, 2.6, there's a significant increase in firmness. So there's a threshold below which fat content has an important impact on the structure of the yogurt matrix and other properties. And in the bottom, we have the same thing with behavior added on the basis of storage time. We also see in the yogurts where there are higher fat contents, they evolve in firmness the longer they're stored. The structure reorganizes and consolidates. It's amplified. And you can see after 3 days, there's a significant increase of firmness. Now, as part of another project, we looked at the cooling rate. Again, we were at 14% total solids, but with two fat contents, 0 and 2.6%. The result was interesting here. When we look, if we compare here the yogurt was put in a container, and it's the time to go from 20 to 4 degrees, either in a room where there was controlled cooling over 17 hours, or a very quick cooling in 3 hours only. So the idea was to simulate what would happen depending on the speeds or quantities of yogurts. What we see is in low-fat yogurt, in blue here, there's no impact on firmness or viscosity as far as the cooling rate. Fast or slow, we get the same properties. However, if we have a 2.6 fat yogurt, firmness is higher and viscosity is as well when cooling is slow. So when there is a fat in the structure, the way the yogurt restructures during the cooling is affected. It doesn't take place in the same way during cooling. Now, again, as we attempted to understand the impact of formulation, the composition of milk, and particularly the resistance to the stirring process, Audrey Gibel's doctoral project studied the casein and serric protein ratio, comparing 3 ratios, 2.81, which is frequently used in industry here, 151, and 3.91, which is similar to milk in which the composition was not modified by the addition of aceric protein. Again, with a test bench to study the effects of stirring and controlling shearing. These results show, and you can see the impact of the ratio on firmness, depending on, and here on the basis of storage time. And here we have day one. You can see there's a ratio effect, a slight increase that is significant between 3.9 and 2.8. So if you add a little whey protein, and when you go have a ratio of 1.5, there's a significant increase of firmness. And restructure over time is affected by the ratio, the protein ratio. So at 1.9, we get a yogurt that after 23 days that it's very firm. The ratio is affected differently by the smoothing temperature. And firmness when we smooth at 25 is higher than at 15 degrees. And here we have the results that I've already showed from Valerie's study. This gives us the same behavior what was done for 2.8. Audrey's work was intended to make the connection with the structure of yogurts varying according to the ratio. We use a very simple method. You don't see the scale, but it's the equivalent of 250 microns. This is an optic representation of a microscopic sample. We measured the surface of the aggregates, and you can see that the gel heterogeneity is different. Here we have microgel particles with larger surfaces here, and the surfaces respond in the same way as firmness. So what about water retention? We used a new method called RMN, a nuclear magnetic resonance. This enabled us to excite the molecules, and before returning to their positions, they will do so at different relaxation times, depending on whether they're very bonded to the protein or in a system with certain mobility, or if they're completely out of phase, which corresponds to syneresis. And we can also look at the intensity of the signal. What I'm presenting here is the yogurt for the three ratios. And if we look T to 4, we have the peaks, which correlate well with, there are behaviors that are different, but there's an increase over time for the 1.5 ratio in particular, some stability for 2.8. And here we look at the T to 3 in terms of dephasing. The distance on the x-axis, we see an increase on the basis of the ratio as it goes up, and a larger temperature effect. To correlate this with syneresis, this method enables us to predict or to measure using a method that does not break the gel in a little tube, to correlate, to do a correlation with what we get as far as syneresis values. And the tendencies are the same. So, the conclusions. We saw the effect of composition, the impact of fat, the optimal cooling temperature. The smoothing is critical for yogurt properties. There's cooling temperature, and there's evolution during storage. And the impact on restructuring during the rebodying phase. My time is almost up, so my time is short. let me talk about benefits briefly. Well, a number of our students are actively working in the field. What's interesting is we've developed methodologies and developed a test beds. The samples that we used were model samples. We didn't use stabilizers. We didn't use the same strains that the various companies might be using. Now, it would be interesting to work with the various companies to use this technical scale unit to see what's happening with their own processes. And we developed water mobility techniques, very interesting for future research and product characterizations. We're certainly open to discussions on that point. We didn't present it here, but in the poster, you'll see that we use the buzz method and the funnel method that give very interesting characteristics that are representative. They're very easy to use. They don't require sophisticated equipment, but enable us to characterize the properties of yogurt. So for us, these are significant benefits that we would be delighted to share. So in conclusion, all of this was the result of work by a team. Here are the students, and we have our research professionals, the teams of Sébastien Villeneuve and Daniel Saint-Gelais.